All right, good to see you. Hope y'all had a good spring break. The highlights of my spring break, I got to go to a Philadelphia 76ers home game. We were visiting some family, and they were behind the entire game and shot a basket with 1 second to go ahead and win the game. So like place went bananas. So that was fun. I hope you had a little chance to recharge. Unfortunately, we do have an exam in 1 week. So sorry to not really easing you back into it very well. Definitely covering the viruses, chapter 12, antimicrobial methods and chemicals. We're going to finish up. I hope we get through antibiotics today, and then we'll see how far we go into on Wednesday. So I was hoping to get through that entire chapter. Again, we'll see where we're at, and I'll make some cutoff on Wednesday about that. I know that having a Monday exam, several of you have said I have my nursing interview on Monday. So I have already rescheduled that with a few of you, but talk to me in the next couple of days and we'll find a different time for you to take the exam if that's a conflict for you. So, all right, so let's jump right into it. We had started, don't be afraid to stop if you have questions, comments, but we'll jump right back into our mini pharmaceutical course here. We talked about a lot of cell wall inhibitors already. That includes the beta-lactams and some others. So we'll jump into our next big group of antibiotics, of protein synthesis inhibitors. Okay, so in general, what can we say about these? There's several different kinds, and remember, as we finished last time, I said, hey, here's a concept map. This is a great way if you're a visual learner to kind of try to help yourself keep all this straight. So you can see here, there's several different kinds of protein synthesis inhibitors that we're gonna talk about next. What can we say about these in general? Most of them are pretty broad spectrum, meaning they're not just working on gram-positive bacteria or not just working on gram-negative, but kind of a mix of both. And some of them work on acid-fast bacteria as well. So we'll get to that. We talked about how there's different fungi and different bacteria that naturally produce antibiotics. This bacteria, Streptomyces, that's the one that kind of acts like a fungi because it grows in filaments and has reproductive spores. Many different species of Streptomyces produce lots of different antibiotics in this class. That's sort of just kind of interesting information there, though. Okay, so aminoglycosides. This is our first big group. They bind to the small subunit of the ribosome. Okay, and remember, bacterial ribosomes, slightly different structure than our eukaryotic ribosomes. So that's where that selective toxicity comes from. The aminoglycosides don't absorb very well through the small intestine, and they are deactivated by stomach acid. So for that reason, the aminoglycosides have to be injected either by IV or intramuscularly. And in general, we have a little more toxicity concerns with the protein synthesis inhibitors in general and aminoglycosides as well. What kind of toxicity? I found this little mnemonic. It's supposed to be a kidney beating up an ear. Okay, so ototoxicity and nephrotoxicity, toxicity to the kidney, toxicity to the ears. And in print, it's kind of hard to see, but printed on those boxing gloves are the names of different aminoglycosides. So we'll explore that a little bit, especially the ototoxicity. Some examples of aminoglycosides that you might have heard of: kanamycin, gentamicin, streptomycin. We're going to look at that one a little bit more closely next. Neomycin, that one is famous for being the second in the triple antibiotic cream. Okay, do you remember what the first one was that we talked about last time? Bacitracin, yes. So that was the first one. And neomycin, I'm not going to mention anymore other than here. So you've all probably used neomycin at some point. So we've got a cell wall inhibitor and then a protein synthesis inhibitor. The third one is a different mechanism. Notice that all these drugs, they end in myosin. Okay, so that's kind of a tip off that that's an aminoglycoside. The gentamicin, okay, why did they spell that one different? They spelled it with an I instead of a Y. It's because that's made by a different organism. It's not made by a Streptomyces. It's made by a different organism. So just FYI, in case you were wondering there. All right, so let's take a little closer look at streptomycin. Here's the structure of streptomycin. Again, I wouldn't expect you to have that memorized, but hopefully you'll notice these are sugars. Okay, sometimes we draw the sugars. That's more closely reflective of how they're actually shaped instead of, you know, drawing them in a perfect ring. So aminoglycoside, an amino modified glycosidic sugar, right? So here's the amino groups here. So amino modified glycosidic sugar. You should be able to look at that and think, oh, that's probably an aminoglycoside. You might not know which one. Okay, so streptomycin was the first aminoglycoside that was discovered. It was first discovered in 1943. We talked about the discoverer of that last time, Selman Waxman, soil scientist at Rutgers University. Actually, it was somebody in his lab, A.I. Schatz was his name, and Waxman was the PI there and just kind of took credit for it. So we could argue about who really did it, but Waxman has traditionally gotten the credit for that. It changes the shape of the 30S subunit. So again, small subunit down here binds to that small subunit, changes the shape a little bit. So during translation of the messenger RNA into proteins, that messenger RNA isn't read quite correctly, and we don't get a usable, we get incorrect amino acids and a non-functional protein from, again, the bacteria. Broad spectrum drug toxicity to the ears. So those of you in anatomy, it actually, streptomycin binds to cranial nerve, let's see, number 8, the vestibular portion of cranial nerve, number 8. Okay, so again, I mentioned that because I know if you're in anatomy, that might make some sense. So what kind of effects does that have? For some patients, it's just a mild ringing in the ears when they take streptomycin, some tinnitus. They might have a little vertigo or they get kind of nauseous. They can have a little bit of hearing loss temporarily, all the way to deafness permanent. Okay, so there's a wide range of effects that can have on the cranial nerve there. So that's why we have to be really careful when we administer this. But when this came out in this probably was, I think it wasn't approved till the late 1940s. This was a miracle drug because this worked, it was the first drug. We had penicillin that worked on some gram-positive infections, but this was the first drug that worked on other very serious infections like tuberculosis, bubonic plague. We were not having as much problem with bubonic plague by improvements with hygiene. Bubonic plague is carried by a bacteria that rides on the fleas of rats. But again, now we have a drug that was useful against that. Tularemia is a drug that, again, it's a bacterial infection that's carried by the fleas on rabbits. So may or may not have heard of that one. That's not common. But so anyway, miracle drug. We don't use this very much anymore because obviously the problems with the ear toxicity, but if nothing else works, sometimes we'll try it. Other protein synthesis inhibitors, tetracyclines. There's actually a whole class of drugs called tetracyclines. They all have 4 rings. Okay, tetracycline. Oh, makes sense. The structure just makes sense. This also targets the small subunit of the ribosome, and this picture shows that. So here we've got tetracycline binding right to that A site, that acceptor site, where the new charged transfer RNAs are coming in, bringing in the new amino acid to add onto the growing peptide. So if tetracycline binds right there, basically it halts protein synthesis. So we would say that this drug is it's not killing the organism, but it is keeping it from growing. It's keeping it from making any new protein. So sometimes we say it's bacterial static. That's usually good enough to give our immune system kind of a little bit of a head start. Another broad spectrum drug, very cheap. Okay, so right from the get-go, we're able to produce that really inexpensively, and that's primarily why this has been an antibiotic that's been used extensively in agriculture. For years and years, been added to chicken feed. I believe that's, I know it's outlawed in California. I believe in other states, there might be some legislation against doing that. But we now know that salmonella that normally lives in the guts of chickens, most strains of salmonella are resistant to tetracycline. Okay, so there's a big push to not use antibiotics in farm animal feed anymore. Why would we put them in animal feed? Because then it keeps the animals from getting sick, and they actually grow faster when they're on low doses of antibiotics. Why is that? We just kind of discovered that by accident. Probably because it cuts down on the normal microbiota of bacteria in the animal's guts, and then those bacteria are stealing nutrients from the animal, so then perhaps the animal grows faster. This is a drug that can be given orally, okay, so pill form. Has a very distinct side effect though. It affects growing bone and teeth. So here's some discoloration that it's caused. So for that reason, we don't ever give tetracyclines so to children or pregnant women, but not to children. Okay, it's fine for adults. Once we're full grown, you may have taken a tetracycline drug, but it does, as adults, we do have to be careful because it does have a phototoxicity. So what that means is patients that are on tetracycline, they can become sunburned much more quickly. Okay, so it's recommended whenever you go outside and you're on a tetracycline that you lather up with sunscreen. Very, very common, used as an anti-acne medication. It has been used for gonorrhea. I don't believe right now it is. That's not the recommendation for gonorrhea right now. In the past, it has been pneumonia, like doxycycline. That's a tetracycline, commonly used. Okay. Okay. And our last class, there are others out there. The last one that we'll talk about are the macrolides. This class of drugs is the other two that we talked about target the small subunit of the ribosome. This one actually binds to the large subunit of the ribosome. Don't worry about these lincosamides. We're not going to worry about those. So it's basically a block, and so that messenger RNA can't pull through the ribosome, and again, that puts a stop to protein synthesis. There's a couple different macrolides that you may have heard of in this class, that you may have heard of. Azithromycin is very commonly used now for things like strep throat and ear infections. Again, right now, we've stopped using this for gonorrhea. Gonorrhea is an infection that if you follow the antibiotics that we've used over the years to that, we have to keep switching because the gonorrhea is becoming resistant to it, or the bacteria that causes gonorrhea. We don't use azithromycin anymore because we don't want strains of gonorrhea to become resistant to that. We want to be able to hold on to this drug and to use it for those, you know, childhood maladies, that we want something that's very safe and can easily be used for that. Oftentimes, if a patient's allergic to penicillin, that's when the doctor will prescribe, it's also called Zithromax or a Z-Pak. And this drug is really kind of nice because we can take it orally and you only have to take it once a day. It comes in a pack of 5 pills, take 1 pill a day. So it's really kind of dummy proof, right, to take it. Why can we get away with that? It has a very long half-life. In other words, it stays up, it keeps its concentration in the tissues. The doses are maintained in the tissues for a long time. So the dosage there is kind of nice. And again, it can be given orally, so that's really nice. Some allergies, not as many allergies as what we see is to penicillin. Some GI issues. The drug that kind of we used before azithromycin was called erythromycin. I used to have to take that when I was a kid, and what kind of GI issues, it made you feel like you couldn't eat, like your stomach cramped up and nothing sounded good. So azithromycin is a little bit better than what erythromycin was in that respect, but sometimes there are some issues there. Okay, so moving on, we have one drug that's a DNA replication inhibitor and one drug that's an RNA transcription inhibitor. Rifampin, or sometimes it's called rifampicin, it's the same thing. Its mechanism is that it blocks RNA polymerase. So remember, that's the enzyme that moves along the DNA to transcribe the DNA into RNA. Complicated structure, I don't really have anything in particular to point out there. It's a semi-synthetic drug. So a lot of our drugs, remember, we said are fully biosynthesized by organisms. This one, I don't know what part of it is added in the lab. Some side chain here or there, I'm not sure what. It's easily absorbed, so we can give this one orally or by injection. It does have the disconcerting side effect of turning bodily fluids kind of a reddish orange color. Why does it do that? Because the drug is that color. You see it in pills, it's like this dark orange color. So I didn't post this, but here's a picture of a patient's catheter bag. Okay, so again, kind of disconcerting, that color, but apparently that it's not anything dangerous or harmful by that. Patients do often experience some nausea, maybe some vomiting, some loss of appetite. So it does have some side effects that we're typically not using this drug for, run-of-the-mill childhood illnesses. Primarily, we use this drug for tuberculosis, along with, remember, the other drug, isoniazid, which was the mycolic acid inhibitor, so TB and leprosy. Okay, so we kind of save this drug so that hopefully it can at least be effective for that TB, which we don't have a lot of options for. Okay, and the DNA, there's a couple DNA synthesis inhibitors, the quinolones and the fluoroquinolones. So quinolones, these were kind of what we might say the first generation drug. Came out with these, oh gosh, and I think in the early 60s when we came out with the quinolones. Nalidixic acid is an example. All right, so I mentioned that because if you were in lab, if you had it already today, we had a media, the CNA blood agar. CNA stands for colistin and nalidixic acid. Okay, nalidixic acid is a quinolone, and it targets DNA gyrase. So if you remember, if here's the replication fork, DNA gyrase works upstream of that replication fork to relax the supercoils. The more and more you open up that replication fork, the more the DNA gets like super twisted up. So it cuts the DNA gyrase, cuts those DNA strands, unwinds it, and seals it back together again. Okay, so both the quinolones and fluoroquinolones target that DNA gyrase. Fluoroquinolones are kind of the second generation quinolones, where we just made the slight change to that quinolone structure and added this fluorine atom. And that one slight change has just made the quinolone drugs way better. Like they have better binding to DNA gyrase, and they work against a broader spectrum of organisms. There are some side effects, so that's one problem that we'll talk about here. Oh, it also improved the penetration into the cell. So for that reason, the quinolones pretty much don't use those anymore, maybe outside of the microbiology lab in terms of prescribing for adults. The quinolones were pretty narrow spectrum. They only worked on against gram negatives. Apparently, the DNA gyrase is slightly different between gram positives and gram negatives, different enough to capitalize on there. Where the fluoroquinolones, again, are more broader spectrum. They're working against gram positives and gram negatives. So they're absorbed well, they're stable in acid. There's kind of certain infections that we tend to use these more for. We kind of reserve them for more serious infections, urinary tract infections that maybe haven't responded well to anything else, some sexually transmitted diseases, anthrax, serious respiratory infections, again, that have not responded to other kinds of treatment. The reason that we save the fluoroquinolones for more serious infections is that it has some potentially serious side effects. And one kind of unusual one is tendon disruption. And this is fairly rare. It most often happens in adults that are older than 60, but it can cause their Achilles tendon to rupture. So that's kind of a strange side effect of an antibiotic, certainly. We think it might limit the blood flow to joints, or maybe it has something with collagen formation. We're not really sure. But those are pretty serious side effects. And when you use this in infants, it has caused seizures or nerve damage. So we don't give fluoroquinolones to children, and in general, don't give them unless, again, other things haven't worked. Okay, aortic rupture, right? That would be bad, right? So we, again, potentially serious side effects there. Okay. Oh, and every once in a while, like I'll come across a mnemonic and I'll pass it on to you. So this is supposed to help you remember the mechanism of the quinolones and the fluoroquinolones. The queen, or quinolone, stops the gyrating dancers. Okay, DNA gyrase. Okay, so I don't know. Maybe that's helpful, maybe not. Okay. So just a couple more mechanisms. Cell membrane inhibitors. We only have a couple of those. If you look at, and they're the polymyxins, colistin, that's also known as polymyxin E. And I mentioned that one because, again, if you're in lab, our media had the colistin and the nalidixic acid. So these are active, especially against gram negatives, because they insert in membranes. And of course, Gram-negative bacteria have the outer membrane, you have a thin layer of peptidoglycan, and then a plasma membrane. So these drugs not only insert in the outer membrane, but kind of poke through that thin layer of peptidoglycan into the plasma membrane. And you see this kind of circular structure here and the tail. That maybe kind of reminds you of a detergent or a soap. Now we talked about soaps and detergent will kind of poke through membranes. Have that hydrophobic head and hydrophilic tail. Did I say that right? Hydrophobic tail, hydrophilic head. Okay. So administration here is usually topical because they have huge toxicity issues with the kidney. Okay, so on rare occasions, they might administer this drug by injection to a patient, maybe again, if nothing else is working, or even like I've read about cystic fibrosis patients getting an aerosol of polymyxin and breathing it in. But again, because of the toxicity issue here, usually just topical, okay, we'll say rarely by injection. Okay. So polymyxin B, I got that right. Yep, that's the third antibiotic in triple antibiotic cream. Okay, so we got the cell wall inhibitor, the protein synthesis inhibitor, neomycin, and now polymyxin B, which is a cell membrane inhibitor. All right, so again, coming at or some kind of infectious organism on your skin from lots of different directions. Okay. All right, I know I'm throwing a lot of information at you. Our last antibiotic that I want to talk about are the sulfa drugs. And they are considered metabolic pathway inhibitors. So they inhibit the metabolic pathway that bacteria have that we actually don't have. And that's folic acid, the pathway to synthesize folic acid. So we'll talk about why we need folic acid and why bacteria need folic acid in a minute. But the sulfonamides, this is a huge group of drugs that have this sulfonamide functional group right there. So like sulfamethoxazole, there's lots of different sulfa drugs, but this works. If you look at the pathway to make folic acid, and don't memorize this, tetrafolic acid, same thing, folic acid, same thing. The substrate here is para-aminobenzoic acid, or PABA. And there's actually 3 different enzymes that bacteria need to be able to synthesize folic acid. The sulfonamide drugs inhibit that first enzyme. There's actually another enzyme here that we didn't name. And another drug called trimethoprim inhibits that third enzyme. Okay, so I don't have those enzymes memorized, don't memorize that. But we often use the sulfa drugs like sulfamethoxazole along with trimethoprim because they work, you could say, synergistically. So if you use those drugs together, they're both targeting the same pathway. They're just making sure that no folic acid is made. Okay, so when we put those 2 drugs together, we call it SXT. So there's the S, there's the X, and there's the T up there. Okay, so SXT, that was one of the drugs that we talked about doing or using for the Kirby-Bauer test in lab. The trade name of SXT, or the name that pharmaceutical companies use when they make it, is Bactrim. So that may be a little bit familiar. Not bacitracin, that's something different. Bactrim is the sulfa drug. Okay, so if we look a little bit more closely, here's just another way a little bit more visual way with the structures, how this drug works. Here's the substrate that first enzyme in the folic acid biosynthetic pathway works on. So PABA, P-A-B-A. And then here's some different sulfa drugs. You can see they all have in common that same structure as PABA. That's the part of the substrate that fits into the active site of the enzyme. So normally we get that substrate, it fits into the active site of that first enzyme. And what we eventually get is folic acid. Not directly, there's just a number of steps there, but we eventually get folic acid. And what's the so what about folic acid? Why do bacteria need folic acid? Well, here they show it right here. That folic acid is necessary in order to make DNA and RNA. Folic acid is a necessary coenzyme, so it has to bind to some of the enzymes that are necessary for replication and transcription, and kind of lots of other things just happens better during replication and transcription when folic acid is present. The DNA is, if folic acid is limited, the DNA is less stable. Sometimes uracil would incorporate into DNA if there's no folic acid. Sometimes we get double-stranded breaks without folic acid. So there's a number of things that folic acid is necessary to make DNA. So what happens if one of the sulfa drugs is going to bind to that enzyme instead? Well, then para-aminobenzoic acid cannot bind to that active site. Say this is one of the sulfa drugs that's binding there instead. And then we get no folic acid as a result. So, I remember what I was trying to think I was going to say. So this is really essentially what kind of inhibition is this? Right, competitive inhibition. Okay, because that drug is binding to the active site of that enzyme necessary to make folic acid and making it so the substrate cannot bind. Okay, so active sites. So I kind of glossed over this. I kind of alluded to it. Why is it that sulfa drugs are selectively toxic? Okay, we need folic acid just like bacteria do. Where do humans get their folic acid? From whatever we eat. Okay, so humans cannot make folic acid. Okay, we don't have those enzymes. We don't have that pathway to make it. Humans get it from food. Okay, so pregnant women, they want to just make sure that they're getting enough folic acid to make sure those cells of the fetus that are rapidly dividing are less chance of having mutations if we've got plenty of folic acid. Okay, just kind of tie things up. Remember, we talked about, those people in history, Selman Waxman, Gerard Domak was the one that developed sulfa drugs in the 1930s. They're pretty broad spectrum, not in use a ton today, primarily because there's a fairly high incidence of allergies. Probably on par, if not more so than penicillin allergies. So we just have a lot of other drugs that are just as effective and again, less incidence of allergies today. Still used for certain infections, still used in things like vet medicine, urinary tract infections, acne, This is one of those drugs that may have a little activity against another microbe, but not just bacteria. Apparently there's some protozoan infections. And I was going to mention, and I forgot, when we talked about erythromycin, that some of you have probably taken it, but that drug was floated back in the beginning of COVID as using that antibiotic to treat COVID. And of course, COVID's a virus, like, we can use an antibiotic against viruses, but it turns out that azithromycin has some kind of anti-inflammatory properties, as well as kind of it encourages the immune system to make certain signals. So they thought that it might have some use, but it turned out that they tested it pretty thoroughly and found out that it didn't. But, so again, some uses, again, not a lot of prescribing of sulfa drugs, as far as I'm aware. Okay. All right. So at some point, I didn't post the concept map that was all filled in, but that's a run through of the drugs that I want you to be familiar with. And it's a good way to just practice, cover up that concept map and try to write it out yourself. Okay. Just kind of do it over and over again. And to me, I think that's maybe more helpful than flashcards in terms of really getting those mechanisms down. You could add in some more detail, some side effects or things like that would probably be the next most important thing. I do also want to briefly mention, okay, well, we focus a lot on bacteria in this class. What about other microbes? What about fungi that cause infection? What about viruses? What about protozoa that cause infection? So let's briefly think about a few other drugs to treat other kinds of microbes and not just bacteria. The main problem with treating these kinds of infections that are not bacterial, these are eukaryotic cells. Okay, so we have issues with selective toxicity. Okay, so difficult to achieve selective toxicity. Remember what I mean by that? We're a drug that targets the pathogen, the disease-causing organism, and doesn't harm the host cells. Okay, so these are eukaryotes. Fungi are eukaryotes. Protozoa are eukaryotes. Viruses, even worse, they're hiding out inside eukaryotic cells. Okay, so we have to have some different strategies to try to get to them. And in general, we have way less options for treatment. Okay. I won't speak very much about antivirals right now. Later on in the semester, when we're talking about some specific infections like influenza and HIV and COVID a little bit, we'll mention some specific drugs. We have mentioned already when we were talking about mutation about some nucleoside analogs. So we have had some success. Drugs that, again, their structure is really similar to a nucleoside. Viruses, which are a little bit less persnickety about nucleotides that they incorporate in. They'll incorporate these nucleosides that are not quite right, and then they'll have lots of mutations because of that, or they'll stop replication because of that. So there are a number of drugs that act that way. And again, we'll talk about that and some other different kinds that have some limited success against viral infections. A fungal infection, most commonly the drugs, there are many different drugs, most commonly actually targeting the cell membrane. Now, our membranes, as well as fungal membranes, have sterols in them. And sterols are necessary to maintain membrane fluidity. Okay, so we always mostly think about cholesterol as being a bad thing, that we get too much of it, but we need cholesterol to maintain our membrane fluidity. Fungi don't have cholesterol. They have something called ergosterol, which is slightly different. And so a couple of drugs like the imadazoles and the triazole, both of these drugs are targeting ergosterol synthesis. So that's kind of the most common mechanism. There are other drugs that actually bind to the ergosterol when it's already made in the membrane and poke holes in the membrane. But I thought, well, let's mention one example at least. In terms of anti-protozoan drugs, oftentimes they're anti-metabolites, so they're trying to capitalize on some metabolism difference that the protozoans have that our cells don't, maybe binding to some aspect of their electron transport chain, for example, or their folic acid synthesis. I thought that I would mention one, since we've talked about malaria a little bit. The most common drug used to treat malaria is artemisinin. It's not the most famous one. Probably you've heard about quinine to treat malaria. Quinine has a lot of side effects, so we've actually discontinued the use of that. And artemisinin, I think that came on the market in like 2016, something like that. And interestingly enough, this is derived from a plant. Okay, so it's not synthesized by other microbes, it's derived from a plant. And this was an interesting article that I read. You can look up sometime if you want. It's an article where they're trying to synthesize this drug in the lab, and just so that they have a little more control over it and they can produce it a little bit more easily. How does artemisinin work? Remember, the causative agent of malaria is not mosquitoes, it's that Plasmodium protozoa. We don't know exactly how it works. We think it has something to do that it causes the malaria Plasmodium to make reactive oxygen species. And that's going to damage some protozoal proteins. All right, so when we'll write that, when metabolized by Plasmodium, reactive oxygen species are produced. So by reactive oxygen species, I mean things like superoxide or some kind of peroxide or something. And that is going to damage the protozoan proteins. Okay, essential Plasmodium proteins. Okay, so that's maybe not terribly satisfying exactly how that works, but has something to do, we think, with reactive oxygen species, oxidizing molecules like proteins that are making them non-functional, basically. Okay, so our discussion wouldn't be complete unless we talked a little bit about resistance. Because before the COVID pandemic, this was, if you asked pretty much any healthcare worker, what's the biggest healthcare emergency, they would probably say antibiotic resistant bacteria. If we would, this is something that's been growing over the years. I can remember back in the early 90s, I worked in a lab that talked about bacteria that were studied bacteria resistance. And I thought, what do you mean bacteria are resistant to antibiotics? Well, we know a lot more about it now. And again, if we'd ever get to the point where, we would get a bacterial infection and not have any drug that was useful, we'd be going back 100 years, and we don't want to do that, obviously. So a little bit, what we do understand. How do bacteria become resistant? Maybe in a population of bacteria, here, maybe a couple, of course, bacteria reproduce really quickly. And if you have a little point mutation here or there that would actually be advantageous, maybe that it made a few cells resistant to some antibiotic. And then if they were in the presence of the antibiotic, so like places like hospitals where almost all the patients are on some kind of antibiotic, if you have that selective pressure, we would say, then the cells that are not antibiotic resistant would die, and the ones that are now have the chance to proliferate, where before they might have been kind of outcompeted if they weren't in the presence of a drug. Now they proliferate to the point where that's really the only cells that are there anymore. So that's kind of the overview of how we think it happens. The specific mechanisms, we're going to get to that in a minute. Drug-resistant organisms, we usually name, it's kind of an alphabet soup of naming. So we talked about MRSA, methicillin-resistant Staph aureus. Here's another example, VRE, vancomycin-resistant enterococci. That kind of list of alphabetic, again, soup goes on and on. How do bacteria acquire this resistance? Well, sometimes it might be like a simple point mutation, like I just talked about. Sometimes bacteria become resistant because they have acquired the ability to make a certain enzyme. And that's not going to be just a simple point mutation. There would have to be, a long stretch of DNA that was just right, that would get transcribed and translated into the right protein. So oftentimes we think bacteria gain the ability to be resistant by some kind of horizontal gene transfer. So remember the three kinds of horizontal gene transfer. Transformation is when a bacteria cell dies, and when its cell wall breaks down, some of its DNA kind of floats out and can get taken up by another cell. Transduction is when part of one cell's genome is packaged into a virus, and that bacteriophage lands on another bacteria cell, and then that part of that genome incorporates into that new cell's genome. But we think most often bacteria acquire resistance by conjugation. And remember, conjugation is when a pore opens up, a pilus is extended to another cell, and the two cells are pulled close together, a pore opens up, and part of a plasmid usually is transferred into that new cell. So lots of information can be exchanged that way. So by conjugation, especially, that's probably more common. What kind of things contribute to resistance? Well, healthcare overuse and misuse. I think this happens a lot less now than maybe what it did 10 or 20 or 30 years ago. Doctors are not going to prescribe something for an ailment that they know is not bacterial, at least hopefully, at least ethically, they wouldn't do that. So we're kind of understanding that we should only use antibiotics when we absolutely need to. Sometimes it's not the doctor's fault, but it's the patient. Maybe they don't take all of their antibiotic. I know that my dad does this all the time. He saves a little for the next time he gets sick. And then maybe you're not getting rid of all the organisms that are causing the infection, and maybe they will come back stronger. For years, we've overused them in agriculture. Profit margin is slim. It's a tough problem. It has to be regulated, really. You can't, it's hard to have, just one farmer say, well, I'm not going to do that anymore, because then that's really going to affect their profits. Worldwide, a lot of different countries, antibiotics are sold as over the counter. So patients, take them when they don't need them, and they don't take the right dose. And we think that that may contribute to the problems that we're having. So to avoid it, whenever you're prescribed an antibiotic, take the whole prescription, unless you're having some adverse effect. Okay, then call your doctor and stop, and maybe they can get you something else. Only take antibiotics when you have to. Saying that, if you're sick and your doctor tells you to take it, then take it. Like, don't be a martyr. But there's some infections, like ear infections, we know almost always clear up on their own. It's a tough one, because that's painful. But maybe there are times where we don't really have to use antibiotics. We often treat serious infections with several drugs, so that in case, the bacteria might become a little resistant to one, we would have another one that worked a different way to knock it out. And of course, lots of research going on to try to extend the life of drugs that we have by maybe just monkeying with a side chain or another, or designing drugs to work against just one specific thing that one bacteria has. A number of years ago, we had a researcher come and give a talk. She did research on Mycobacterium tuberculosis, and they found one protein that only that organism made, and they were trying to develop a drug to just that one protein in that one cell. So it's tough, though, because antibiotic development is not lucrative. Pharmaceutical companies would much rather make a drug that you have to be on the rest of your life, rather than just some to 10 days. So it's a tough problem. So we look at specifically kind of what's going on at the molecular level that allows a bacteria to survive in the presence of an antibiotic. And this is a little bit more, probably the most complicated part of all of what we've talked about on antibiotics. So there are some homework questions about this. Don't be afraid to go back in and read that section. I think it's 13.5 in your book. It's easy to get mixed up, like who's doing what, basically. So here's some different mechanisms, though. Again, they talk about all of these in the book. Read that over, or come talk to me more about it. We've got all week before the next exam. So first mechanism, bacteria prevent the uptake of a drug. So if a bacteria can, say, maybe alter its porin structure. So remember, the porins, those are the channels in that outer membrane, the kind of, they're transport channels. So if a bacteria, say an E. coli, could maybe, if there's a point mutation or two in that porin structure, maybe that can become more selective, and the antibiotic is not able to gain entrance to the bacterial cell anymore. Okay, so the idea is the less drug inside the cell, the better chance that the bacteria is going to survive the exposure. Along those same lines, there are bacteria that have pumps, so to like get rid of toxins. Pseudomonas is really good at this. That it has these efflux pumps that they use energy, they use ATP, that when they're exposed to some kind of toxin or like an antibiotic, they can just get rid of it right away again. So we think that's one way that cells become multi-drug resistant. Doesn't matter what it is, it just pumps it right back out again. Okay, so preventing the uptake of the drug, that's one mechanism. The second mechanism, we've talked about this already, enzymatic inactivation of the drug. So that means the bacteria are making some enzyme that will render the drug ineffective. Okay, so an example of that is beta-lactamase. Okay, so some bacteria make this enzyme, beta-lactamase, that acts on some beta-lactam drug and cleaves that beta-lactam ring, opens it up. So now the drug doesn't bind to that transpeptidase enzyme in the bacteria anymore that would keep it from making an intact cell wall, basically. Okay, so That's one example of an enzyme that bacteria make to deactivate the drug. I searched the literature for some other examples. There are other drugs. So here's a drug, gentamicin. They found some bacteria that are able to live in the presence of gentamicin because they're able to just slightly change some functional group of the antibiotic. So in this case, I think it said they were able to phosphorylate these hydroxyl groups here that just made it so that gentamicin didn't bind quite in the same place to the small subunit of the ribosome, wasn't able, affected the binding. Okay, so altered the drug, so wasn't able to bind. Sometimes the bacteria can modify itself. So rather than changing the drug, it can modify itself so the drug can't bind. Okay, so here's a couple of examples of that. Some bacteria can methylate their ribosomal RNA. So add a methyl group. So that's just a CH3 group. They acquire the ability, again, to make these methyl transferases that will just add a methyl group to some of their ribosomal RNA, usually in the small subunit. And then basically drug can't bind. We'll just say to the ribosome. Okay, so it doesn't affect the function of the ribosome or the protein synthesis of the bacteria, but then the drug can't bind. And along those same lines, if the antibiotic is targeting an enzyme, might be able, just with a point mutation in that enzyme, in say DNA gyrase or RNA polymerase, that doesn't affect the functionality of the enzyme. It's just then the drug can't bind to it. Okay. And a couple of other mechanisms that are a little bit, I know mechanism 5 was just recently kind of discovered, but mechanism 4, target overproduction or enzymatic bypass. So the bacteria can start to make much more of wherever that antibiotic is targeting. So say it's a target enzyme, say it's RNA polymerase. Now the bacteria starts making many, many, many more copies of RNA polymerase. So, you know, if the drug maybe can't bind to them all, it still has some functional RNA polymerase. Or they've even found instances where the bacteria has figured out a way so it doesn't need RNA polymerase anymore. Or maybe, you know, it can find a way around to reproduce without that enzyme that the drug is targeting. And again, that more recently discovered, there are proteins that mimic the binding site of drugs, so that the real target site is at least some of it left functional, and the antibiotic just targets to these red herrings, I guess you could call it. Okay. All right, so I think, so this is a little exercise. Take a look at, I'm not going to take the time to go through it right now, but basically what I've done is listed some different mechanisms and see if you can figure out which, they're not in the right order, 1, 2, 3, 4, 5. See if we can figure out, for those of you that are, you know, it's nice to have a picture attached to some mechanism and see if you can match those up. Okay, so we'll look at that, and I'll put the answers up at the beginning of next lecture. So look that over and ask me if you have any questions. Okay. Questions or comments about antibiotics, resistance, anything that we've talked about? Okay. I didn't post this picture, but you know, maybe someday we'll be able to make some viruses that work against the bacteria that we want to get rid of. That's sort of the pretext. If you've ever read Sinclair Lewis's Aerosmith, was written in 1925, well before antibiotics were made. And he was trying to cure bubonic plague by using viruses to target the bacteria. So it's kind of, it's a good read someday. Someday put that on your list. Okay, but I do want to move on. Okay, so we're going to move on to talking more about the process of infection. Okay, so hopefully you find this meaningful. Those of you that are going into fields of medicine, we're going to, it's a little bit of terminology, but a lot of terms that I think that you'll be familiar with, kind of walk through the process of infectious disease and think about the kind of repertoire or the arsenal that pathogens have and what makes them successful in causing disease. We call those things virulence factors. And then the next chapter really piggybacks on it and talks a little bit about epidemiology. Okay, so we'll get to that for the next exam. But we'll first start with talking about our normal microbiota. Okay, so we have lots of bacteria, and not just bacteria, but other microbes in our body already that we know are really important in keeping us healthy. We don't understand a lot of it. I read once an author had said, studying our microbiome is not rocket science. It's a lot more complicated. Okay, so we don't really understand, what microbes should be there to make us the most healthy, but we know that we do have a lot of indigenous microorganisms that we call our microbiota. So not just bacteria, but we have archaea as normal, like our microbiota. We have fungi, usually yeasts. I don't know that there's any molds that are normal flora. Yeasts, there's some protozoa, maybe even some viruses that are part of our normal microbiome, and to some extent important for us to be healthy. So we could describe that relationship of those microbes to us and some synergetic or maybe symbiotic relationship, either mutualistic or commensalistic. So a mutualistic relationship is when both organisms benefit. Okay, they're mutual benefits. Or a commensalistic relationship is when one organism benefits and one organism is not affected. I should say impacted, because I always forget how to spell affected correctly. So forgive me if that's wrong. Okay, so lots of examples of mutualistic relationships with bacteria. The bacteria in cows' rumens. The cows actually are not digesting the grass. It's the bacteria that are digesting the grass for the cows. And in turn, the cow gives the bacteria this nice, warm, nutrient-filled environment. A commensalistic relationship is a little bit harder to pin down because a lot of times relationships that we once thought were commensalistic, once we learn more about it, we actually realize that both organisms are benefited. Like the Staph aureus on our skin, it doesn't seem to do us any good, but certainly we're giving those Staph aureus some nutrients and a nice warm place to live. But we now understand that Staph epidermidis, that's what I meant to say, Staph epidermidis, is likely outcompeting other organisms that might be more likely to give us a skin infection. So that's a little bit, again, harder to pin down. We know that some of our microbiome is only transiently associated with us. So maybe, again, this is a temporary association. A lot of some of the microbes, like in our upper respiratory tract, maybe they kind of establish themselves for a while and then they kind of get outcompeted or go away. Some of our microbiome, though, we would consider to be residents, meaning you acquired those bacteria or whatever soon after you were born, or maybe during the birthing process, and pretty much you have them for the rest of your life. And of course, there's a lot we don't understand, like what happens when we go on antibiotics, how soon does it take till we get our kind of normal variability of bacteria back, or if ever do we? So again, there's a lot that we don't understand there. Just sort of kind of, if you're interested, this is just a list of some of the kinds of organisms that we would find in our body. And of course, in different places of our body have a different microbiota, depending on, you know, access to oxygen and all sorts of other things. Our lower digestive tract is essentially not oxygen in there, so we get some more anaerobes down in there. There are parts of our body where there are not any microbiota. We call those parts axenic. So if we're healthy, there shouldn't be anything growing there, like a lot of our internal organs, like our heart. You don't want bacteria in your heart or your central nervous system or your lymph or your blood. Patients that get an infection in their blood, that's called sepsis, and that's a life-threatening condition when you have a blood infection. So, all right. So a few definitions. We want to try to use these terms, try to use them appropriately. If a host is injured by a disease-causing microbe, we call it disease. But just if that microbe has entered and established and multiplied, we would refer to that as an infection. Okay, so an infection doesn't necessarily cause disease. It doesn't necessarily cause damage to the host. It could be asymptomatic for a short period of time or a long period of time. Okay, so like our normal microbiota, sometimes you could think about that as an infection, but we don't usually call it that because it's kind of the normal state of things. Okay, a disease-causing microbe, that's a pathogen. And again, I think I've mentioned this before, but if we use that correctly, a pathogen is a bacteria or a virus. If the disease-causing microbe is a yeast or a protozoa, we would more accurately call that a parasite. I noticed places in your book that hasn't used those two terms accurately. So either it's kind of a loose designation or we just kind of make a mistake very often. But all right, an objective and measurable consequence of infection. We would correctly call that a sign, and then an effect of disease felt by the patient is a symptom. Okay, so again, signs and symptoms, those are terms that, you know, we try to use correctly. Sometimes they aren't always used correctly, but an example of a sign, again, something that a doctor could observe, like a rash or vomiting or diarrhea, fever, blood pressure. Okay, we can measure those things. A symptom then might be some kind of pain or malaise, tiredness. Okay, how do you measure that? It's not easy. Okay, so we'll try to use those words correctly. If we look at the progression of disease in an individual, we like to kind of set that up into different stages. So right here would be when that pathogen first enters, what we call the portal of entry. And we'll talk about different portals of entry soon. But say it enters a human host. Typically, there's going to be a period referred to as an incubation period, where the pathogen is dividing, but there's not enough of it in the host yet to be causing any symptoms, no signs or symptoms. Okay, so no signs or symptoms. Typically, as we increase the number of pathogens, typically inside the host, typically the signs and symptoms then are going to increase. Okay, so after a time and the incubation period could be a day, it could be an hour, it could be 9 months. It can be very different, depends on the infection. And I do talk about a lot of different infections as I'm bringing things up. Worry about the principle, not the specific infection yet at this point. Just like when we talked about viruses, worry about the principle. As the patient starts to have some non-specific symptoms, we call that the prodromal period. Okay, so mild and usually non-specific signs and symptoms. Okay, so you wake up, your throat's a little scratchy. Oh gosh, I think I'm getting something. Okay, or your nose is a little bit runny. Okay, those are the kind of non-specific, a prodromal period. You might be getting a cold, you might be getting the flu, you might be getting COVID, you might be getting anthrax. Like all those things kind of start out the same. And then as the pathogen is replicating and the host is going to start to feel more signs and symptoms. Okay, so most severe signs and symptoms are during what we would call the illness stage. And that might kill the host, and that would be the end of the infection. Or eventually, hopefully, the immune system is going to start kicking in and starting to kind of kill some of the pathogens. So enters into a period of decline. So again, the immune system clearing the pathogen. Signs and symptoms are getting better. Although the host sometimes is going to be susceptible to some secondary infection. So how often maybe does somebody have a cold or the flu and then it turns into pneumonia? Okay, so that original infection has weakened the host a little bit, that they're more susceptible to other infections. That can happen. Again, hopefully if the immune system is winning out, then you would still maybe have a period of convalescence where you have, normal function. And probably very few signs or symptoms, but there's still a potential that there may have been some chronic damage. Okay. Question?
Is there a difference between the blue and the red line? Is it like disease and
That's a good question. Is there a difference between the blue and the red line? Usually for most infections, they're going to be about the same, but there's probably some variation there. So usually the more pathogens present, the worse your signs and symptoms are going to be. And as your immune system starts killing the pathogen, then your signs and symptoms will be less. Now, there's some infections like whooping cough. The signs and symptoms are caused by a toxin. And even though we're getting rid of the organism, that toxin persists for a long time. So we might have a little different curve with an infection like that. And one other thing I'll say here is about when is the patient contagious? Patient can be contagious during any of those periods. Chickenpox, contagious in that incubation period. So you have people walking around, no signs and symptoms, they feel just fine, and they are, can potentially be giving that to other people that are susceptible. COVID, at least in the early mutation or, early forms of COVID, contagious in that incubation period. Some infections that are especially diarrheal disease, they're still contagious during convalescence. Patient can still be shedding in the diarrhea. So, okay, that seems like a good place to stop. We'll continue on Wednesday. Have a good day.